Hello everyone and welcome. My name is Timothy Farren and I'm a clinical scientist in haematology and the head of immunophenotyping at NHS East and South East London Pathology Partnership, which is hosted by Barts Health NHS Trust. I'm also the scientific lead for the SIHMDS and I'm an honorary senior lecturer in hemato-oncology. It gives me a great pleasure to present this presentation on flow cytometry in acute leukemia. And I appreciate that many of you will have varying levels of expertise in flow cytometry, with some having more experience than others. As such, I thought it would be best to give this presentation to split it into two halves. As I'm sure you can appreciate, it's quite a big task to try and accommodate both flow cytometry and acute leukemias into a lecture. So firstly, I will present on the basic principles of flow cytometry and basic plot interpretation. Then the second half will focus on the clinical applications and, where possible, with any correlation for morphological review. So I thought I'd start with a quote that I feel is quite appropriate to share here from Mahatma Gandhi. And Mahatma Gandhi said, "A correct diagnosis is 3/4 the remedy." And I think this is something that really resonates to all pathologists. We all feel that working in an integrated fashion results in the right diagnosis for the right patient at the right time. I think that's really a key theme to remember, that integrated diagnostics is the only way to achieve that right diagnosis. So what I'm going to cover in today's presentation is when to use flow cytometry, what is flow cytometry, the reporting classifications that are out there, and also some of the more recent changes to some of the classifications. I'll talk about integrated diagnostics, some plot interpretation, flow cytometry in acute leukemia, and we'll also go through some worked examples. So when do we consider flow cytometry advantageous in hematological conditions? Well, it's commonly used in acute leukemias, chronic leukemias, lymphomas, plasma cell dyscrasias, myelodysplastic syndrome and other related disorders such as aplastic anemia and PNH. In addition, flow cytometry can be used for assessing prognosis in certain hematological malignancies, but also assessing responses to therapy, looking for what is now coined measurable residual disease, or MRD. Although this is not covered in this presentation, MRD assessment by flow cytometry is increasingly being used in clinical trials as well as endpoint assessments. Immunophenotyping is routinely carried out using blood, bone marrow, lymph node biopsies, CSF, pleural fluid, ascitic fluid, as well as skin and gut. Cytometry is only one small part of the diagnostic process, and not just in acute leukemias, but in all hematological malignancies. The integration starts in the clinic or on the ward with that clinical suspicion, which initiates the diagnostic process. And this includes morphology. Obviously, the morphology of a blood film following a full blood count is one of the key first investigations that is undertaken within the laboratory, which then naturally initiates a cascade of other disciplines and investigations to result in an integrated diagnosis. This includes immunophenotyping or flow cytometry for cellular identification and lineage association. It also includes cytogenetics, including G-banding and FISH. Here we have a gentleman's karyotype with 45 chromosomes present. But also we can see we've got a monosomy 7 there, a loss of one of the chromosomes 7s, and an abnormal chromosome 3. And of course we've got molecular genetics, such as FLT3 and NPM1 mutation screening, as well as next generation sequencing. So really, flow cytometry is only a small part of the integrated diagnostic process, particularly for acute leukemias. So what we need is, first of all, the clinical suspicion. What you see on the ward in the clinic. We want to know about skin rashes, lumps and bumps, the personal medical history of the patient. Then, of course, you've got your baseline testing, including morphology from maybe a full blood count, and your biochemistry and other investigational modalities. Then that sort of will direct the more specialized testing, including immunophenotyping and immunohistochemistry, and of course, molecular genetics and cytogenetics. And in terms of reporting, how do we go about this? Well, we currently report to the WHO World Health Organization 4th edition on the classification of hematopoietic and lymphoid tissues, and this is published in 2017. However, much more recently, 2 new proposed classifications have been published in manuscript format, with the World Health Organization 5th revision published in leukemia, and the International Consensus Classifications published in blood. And these are the references in which they have been published for you to have a look at, and you can download freely from the internet. But as we know, there are inherent problems with many classifications. And this includes where classifications change, and we have seen this quite recently with the 2 new proposed WHO and ICC classifications. Samples can be somewhat unreliable, particularly in some malignancies such as myeloma, where we know that plasma cells will form focal aggregates and aren't easily aspirated, and a trephined biopsy is going to be much more informative. Tests can be unreliable. Pre-analytical and analytical variables can significantly impact the diagnostic outcome. And many specialized tests still require some element of manual preparation, and human error can always impact the testing and analysis. So we can confidently say that no single test will ever provide an answer, and everything must be reported in the clinical context of the patient. So what is flow cytometry, which is sometimes referred to as immunophenotyping or occasionally immunohemato-oncology? In its very basic form, it's the measurement or metry of cells, which is obviously the cyto part, as they move in a stream, or the flow part, past a set of stationary detectors. So essentially, it's the joining of the principles of immunohistochemistry and morphology. It uses the size and the shape of the cells. We look at antigen expression both on the surface and internally to the cell, cytoplasmically, and it utilizes the power of light scatter and fluorescence. So in other words, it analyzes a population of cells on a cell-by-cell basis, a critical capability for today's pathologists and scientists and clinicians who are looking for the very few cells amongst many cells within a sample, very much like finding needles in a haystack. And this will enable them to study a disease or a biological process. These cells are in a suspension which have generally been tagged with a fluorescence dye, which are then forced into a narrow stream. Secondly, flow cytometry is extraordinarily rapid. Routine sample analysis rates can range in excess of 10,000 cells per second, even up to say 30,000 cells per second. And this is an incredible enhancement over historical methods of visually examining and counting cells. And finally, flow cytometry has the capability to simultaneously measure multiple parameters, otherwise known as multiplexing, of these single cells. And multiplexing allows scientists and clinicians to gather more information from a single sample faster than ever before. And it's these capabilities that have made flow cytometry a really powerful tool with multiple applications for pathologists. So there are a number of flow cytometers which are commercially available, and these are just 4 types from some of the 4 larger manufacturers. You can see we've got what's on from BD, Beckman Coulter, from Agilent, and Sysmex. And I think the first thing to say is these 4 systems look relatively similar. They are boxes containing wires and lasers and detectors. And this is this inside of the system. So you can see you've got, this is what they call the flow cell here. You've got these prisms which are sort of focusing the light down into very fine beams. You can see lasers and information being detected within the flow cytometer. And how do these boxes that contain all these wires and lasers and this sort of intricate network of detectors within this system, how do they work? Well, the flow cytometer has several major organs. We've got the fluidic system that funnels the sample of cells, such as the blood or the bone marrow, into a single stream, and this is known as hydrodynamic focusing. So the cells pass through a laser beam one cell at a time. As each cell passes through the beam, it scatters light, and it may also emit fluorescent light. And these signals are collected by the optics or the optical system and routed to various detectors. The signals received by the detectors are then converted into numerical values by the electronic system. These results can then be displayed on a screen or saved for future analysis using specifically designed software. As each cell moves through the beam, its parameters or its characteristics are measured and recorded along with the time that it passes through the beam. And typically data can be collected for at least maybe 10, 20, 30,000 cells in excess of millions of cells per sample. So this is a cartoon characteristics or representation showing a cross-sectional part of a flow cell. So you've got the sample core where the sample goes through in the middle, which is surrounded by sheath fluid, and the outside of the flow cell. So you can see here you've got the sample being forced up here, surrounded by the sheath in this sort of conical funnel. And what you can see here is you've got your cells passing through 3 lasers going across here, and the cells are passing through one cell at a time. And this is that process known as hydrodynamic focusing. What we don't want is this situation on the right here where you've got 2 or 3 cells passing through the laser beams at any one time. This is where you get doublets that are stuck together, and this can cause an erroneous result. So what we want is what we call doublet exclusion. So how does it measure each of these characteristics? Well, you've got your laser beam here, you've got your sample, your particle that's flowing through. If you plot time versus signal intensity, as your sample starts to enter the laser beam, so you get a change in that signal intensity. As the cell is all the way through the middle of the laser, so you get a peak in that signal intensity. And then as the cell passes out the other side, so that signal intensity drops off. And that gives us that accurate enumeration of cells. At the same time, you've got laser light coming across, and you've got light being shone forward. And that is forward scatter, and that tells us about the relative size of the cell. And then at 98 degrees at right angles, we've got fluorescence being given off, and this is what we call side scatter. What this does is this tells us about the cell complexity and organelle content. So when we define populations, we can use the power of forward scatter or size and side scatter, which is internal structure and granularity, to be able to differentiate cells within a heterogeneous cell population. So we've got here the red blood cell, which is our smallest cell, and if we look along the forward scatter, the size axis, we've then got our lymphocyte, followed by a neutrophil, followed by a monocyte. If this is our peripheral blood sample and we're looking at side scatter and we're looking at organelle content, we know that our red cells are the smallest with not much nuclear remnants in the cytoplasm. We've then got the lymphocytes. Occasionally you might get some large granular lymphocytes, followed by your monocytes, with our neutrophils being the most granular of all. So if you plot one against the other, this is what you see on the right hand side here, and you can see that you've got cells that are increasing in size and increasing granularity. And this is exactly what we see on a full blood count printout, and this is from one of ours. You can see here that you've got your red cells and debris falling down here. Your lymphocytes can be gated here, and that's drawing a circle around the cells of interest there, your monocytes and your granulocytes. So it's very easy, just by using these very simple parameters of forward scatter and side scatter, to enumerate a cellular population within a heterogeneous group of cells. So flow cytometry is also truly considered multi-parameter, and this is further enhanced by the staining of cellular populations to clearly define and enumerate them within this heterogeneous population. So you can see here we've got some immunohistochemistry on the left-hand side, and it's been stained, and you can see whatever it is, it's positive, very strongly positive. You've got lots of brown stain going on there. In flow cytometry, we can stain cells with, say, 8, 10, 12, 15, 20 colors at any one time to actually tell us in depth what these cells are. We can actually be very specific, whether it's a T cell, B cell, myeloid cell, and also look at maybe the B cell subsets within the population. So you can see here you've got kappa and kappa on these axes here, and you can actually draw gates around the cells of interest. You can see here we've got 19 and 5. We've got our T cells up here, B cells down here. We've got a co-expression of the two there. So how are we able to enumerate and actually assign a lineage to identify what these cells are within the heterogeneous population? We do this by using commercially bought monoclonal antibodies, seen over here. And these antibodies are attached to a dye or a fluorophore, or sometimes known as a fluorochrome, and these cells are mixed together. So if this was a case of chronic lymphocytic leukemia or monoclonal B-cell lymphocytosis with a CLL-like phenotype, here we've got our CLL cell. We've got CD19 binding to the cell surface. We've got an anti-CD23 also binding to the cell surface, as we know that CD23 is upregulated in CLL. And you can see here we've also got the natural killer cell antigen CD160 and anti-CD160 binding to the cell surface. So we've got a B cell, because it's 19 positive, which is co-expressing 23 and CD160. You can see CD2 over here with a purpley sort of fluorochrome attached to it. As this is a CLL cell, obviously does not express CD2, therefore CD2 will not bind. And therefore, when the cell goes through the flow cytometer, that CD2 won't fluoresce. So how do these antibodies and these fluorochromes, these fluorophores work? Well, it's sort of very basic physics. As the laser that tells us about the forward scatter and the side scatter of the cell interrogates the cell, so the fluorochrome attached to the antibody that has bound to the cell absorbs some of the light. And this becomes excited. So this is what we call an excitation wavelength that sort of activates the dye, and the dye absorbs some of that energy. As those fluorochromes or these fluorophores become so excited, they need to release this energy in a form of a photon of light, and we know the spectral properties of that dye. And we know that specific wavelength, or the emission wavelength, that is given off by that dye, and that is always longer than the excitation wavelength. So by knowing the excitation wavelength and then by knowing the emission wavelengths of these dyes, we can then combine a number of dyes, a number of colors together to be very specific about what these cells are. And like I said, some of these more novel flow cytometers will do 8, 10, 12, 14 colors, and some of the more sort of specialized research instruments could even do up to 21 or even more colors. So this is another sort of cartoon illustration of the sort of multi-parameter processes within flow cytometry for hemato-oncology diagnostics. So we have the cells here. The cells surrounded by sheath fluid are passed through the flow cytometer, or the flow cell, this sort of conical cell, flow cell here. Cells are passing through one at a time, and you can see you've got a variety of cells here. The laser then, of course, comes across, and we get forward scatter telling us about the size of the cell, and side scatter telling us about organelle content and granularity. At the same time, those very same lasers that tell us about size, granularity, as well as some additional lasers to allow us to combine multiple colors together. So those lasers that interrogate the cells excite the fluorophores. Those fluorophores build up the energy and then release their energy in a form of a photon of light in that emission wavelength that is longer than the excitation wavelength. But I think it's we have to be very mindful all the time that flow cytometry is just one test, and it tells us a lot of information, but it won't give you the architectural information, particularly in certain cases such as myelofibrosis, hairy cell leukemia, and multiple myeloma, where we know that plasma cells form focal aggregates within the bone marrow. So it's very important that we remember that what's in our sample tube over here, those cells might not be what is represented in the bone marrow aspirate smear, but also the trophine biopsy. So it's very important that when we look at flow cytometry, we remember that it's part of an integrated process. It's part of the bigger puzzle for a patient's diagnostic outcome. And it's also really important that we remember that when we're reporting flow cytometry, when we're reporting the flow plots, that this is done in conjunction with looking at either the aspirate smear, the full blood count smear, or a cytospin preparation, because you can only really truly interpret it when you've got morphological confirmation alongside. So it's very important that we report them side by side. And then obviously the genetics, the histology, the immunohistochemistry can then follow and support that diagnosis. So what are we really looking for when we're looking at flow cytometry? Well, it's pattern recognition. We know what normal cells express, we know what growth arrested cells look like. So we're looking for the presence or the absence, the upregulation, the downregulation, the overexpression or the underexpression of key antigens within some hematological malignancies. But I think it's very important to remember that obviously every single individual patient is an individual, and as no one patient will behave the same, hence no single diagnostic test will suffice, because there is no real one-size-fits-all approach. So first of all, let's look how we define these populations. I think this is quite important that we understand what these plot interpretation involves. So here we have a dot plot of forward scatter versus side scatter, and we've enumerated our lymphocytes, our monocytes, and our granulocytes. So forward scatter and side scatter, we can then draw gates, and that's physically drawing gates and circles around the populations of interest to give us an enumeration. The other thing we can do is obviously we've added antibodies with fluorochromes or fluorophores attached to them, which are then passing through the flow cytometer. Of course, as that emission wavelength is given out, we're able to detect that within a number of detectors within the flow cytometer. So what we can do, and just for this illustration here, you've got a blue detector versus count. So we've got cells that are negative for whatever is on that blue fluorochrome, and you've got the cells that are positive over here. So this will be our sort of double negative, our negative population. So you've got a bunch of cells that are negative for whatever is blue, and the positive ones over here. And then, like I said before, what we do is we draw a gate around the cells that we're interested in, and that will give us an enumeration of how many cells there are of that expressing that antigen within the heterogeneous population. Likewise, we can then combine, like I said, a number of different colors, a number of fluorochromes together on a number of antibodies to be very specific at pulling out cells of interest. So we've got the blue detector on here, we have the red detector on here. So if we said our red is CD5 over here, so these in our Q1 up here are CD5 positive cells that are negative for everything else. So these are our T cells up here. We've got CD19 on our blue detector down here. So these are our B cells over here, our normal polyclonal B cells, hopefully, that are expressing CD19. So those are our single positive populations of CD5, CD19. We've got our double negative population here in Q3 that's not expressing either, and we can use that to set our gating coordinates. And then over here you've got this single cell up in Q2 that is co-expressing both CD5 and CD19. So when we're looking at plot interpretation, we can look at what we've just said, the mutually exclusive, where you've got T cells and B cells on separate axes. And when we gate on our lymphocytes, we can pull out, and like I said, you can actually enumerate, so we've got some percentages over here of the cells within that heterogeneous population. We've got the co-expression. So on this axis, you've got CD7 and CD3, and these are our T lymphocytes up here co-expressing both of them. And then we've got non-expression. So it's very important to know what you're gating on. So up here it's telling us that this is gated on lymphocytes. We've got CD34, our stem cell factor, on this axis. We've got CD117 or c-Kit on this axis down here. And you can see that there is no expression of either CD34, CD117, or a co-expression of 34117. So this lymphocyte population, you can see that there is no evidence of immaturity with either CD34 or CD117. And if you remember, we also talked about when we're looking at plots and populations of interest, we also like to know about the intensity of some of these populations, because that may tell us about what the hematological malignancy might be. So we have in the first plot over here, you can see here this is a negative expression. Everything is falling down here, which is negative. When we talk about dim expression, we like to see it sort of falling within this first log over here, and you can see this is a dim expression of CD56. So this indeed could be natural killer cells, which we know have a weak CD56 expression. We've then got a moderate expression of CD8 on our T cells over here that's falling sort of between the second and third log decade on this plot here. And then we've got CD38PE, which has got a very bright overexpression on this population down here. So if this was a bone marrow aspirate, we might find that these are our plasma cells over here. So by combining both the intensity of the expression, gating on the populations of interest, and actually physically drawing gates around them, and by plotting on our X and Y axis fluorochromes and antibody targets, we can then enumerate quite accurately the number of cells of a certain population within a heterogeneous group. Something else that can be done by flow cytometry is looking at maturation patterns of certain populations. Now we know as blasts transition to the myeloid lineage, they acquire higher levels of CD13, CD15 and CD33. So the upper panels up here, plots A, B and C, depicts normal myeloid maturation with myeloid elements shown in green. We've also got some monocytic elements, some mature monocytic elements shown in purple over here in panel B for comparison. So it's very important that most flow cytometry labs include CD45 within their gating strategies, and this is usually the very first gating strategy that is undertaken. So you can see here in this first plot we've got CD45 on the x-axis versus side scatter on the y-axis. And what we've done here is we've got CD45 strong gated lymphocytes falling here with a lower side scatter. We've got our monocytes, which have a slightly dimmer CD45 compared to our lymphocytes, but a higher side scatter. We have our myeloid series falling up here with increased side scatter, but again a weaker CD45. We've got a sort of CD45 dim low side scatter window, and then we've got this CD45 negative population down here, which is commonly where our debris might fall and stuff that's not been stained. Now this stuff down here, this debris, it's very important that we don't always exclude that, because we know that malignant plasma cells can fall down here, and we know that pediatric patients with B-cell acute lymphoblastic leukemia will have a very sort of low negative CD45 expression. So even when you can see this nice sort of plots being pulled out here and we can gate on the cells of interest, it's very important that we check this sort of debris area down here for any expression of antibodies. So if this is our normal person on the left-hand side here, you can see on the right-hand side we've got again CD45 plot versus side scatter on the y-axis, And you can see you've got your debris area falling down here. You've got your lymphocytes over here with a stronger CD45 expression. But you can see that you've got a very sort of reduced side scatter and this big sort of mass of cells that are falling in the CD45 dim with a low side scatter population there. So what are these cells? Well, we can draw a gate around them. So we've drawn a great gate around this population down here. And we can then look at the antigen expressions on those cells. This sort of CD45 dim or intermediate area with a low side scatter is sometimes referred to as the blast window. And this is quite commonly where blasts may fall. So it's very important that if we do see a population like we see here on the right, we gate on those cells, and then we can look at the antigen expression profile both on the surface and internally using cytoplasmic techniques to see what that cell is expressing, to identify it. Is it a myeloid cell? Is this a case of acute myeloid leukemia, or is it something else? One of the questions we quite commonly get asked is, well, how do we design our panels? What do we want to include within our panels? And whilst standardization is the optimal goal for flow cytometry, we're still a very long way off standardization for flow cytometry panels. However, I'd like to highlight the Euroflow group because it's a Euroflow consortium have looked at designing specific antibody panels for the diagnosis and classification, but also monitoring of hematological malignancies. And I've got the reference here on the slide, and you can go to the website and you can download copies of the antibody panels that they use. So they've got things such as the LST or the lymphoid screening panel. They've got panels for AML and for MDS. And they've got panels for things such as B-cell acute lymphoblastic leukemia, residual disease monitoring. And this is really sort of the biggest consortium, the biggest approach to standardization that we've seen in flow cytometry. So I'd advise you to have a look, particularly if you're just sort of starting off, to go onto the Euroflow website, have a look at the panels and the plots that they have there, as it might give you an idea about how you want to set up your flow cytometers and your flow cytometry service. So that covers the principles of flow cytometry in clinical practice. And we've covered antibodies, antigens, a little bit about gating strategies and what we're looking for, and how we can enumerate cellular populations and subdivide populations to actually tell us what these key cells are within a heterogeneous group. So you can now pause the presentation, have a quick break, and then when we come back, I'll cover flow cytometry across leukemia, acute leukemia, and look at the interpretation of results, and where possible, any correlation with morphological examination. Welcome back. And so now let's have a look at the role of flow cytometry in acute leukemia. So this table summarizes quite nicely the process of lineage association to progenitor cells for suspected acute leukemia. So in order to assign characterization, we can use lineage-specific antigen targets. So for the myeloid lineage, we want to see an expression of myeloperoxidase, MPO, or monocytic differentiation, where MPO intensity in part exceeds at least 50% of the mature neutrophil population. In terms of monocytic differentiation, 2 or more positive antigen targets are required, such as CD11C, CD14, CD64, or lysozyme. Now for T-cell lineage association, the protein complex and T-cell co-receptor CD3 should be expressed cytoplasmically. Very rarely it can be detected on the surface of the cell, but we certainly have seen cases where it is. It is important that the antibody that is targeting the CD3 is the CD3 epsilon chain. And again, the intensity in part should exceed 50% of the mature T cells by flow cytometry. And then finally, for B cell lineage association, what we like to do is see a strong expression of CD19. However, if we've got a strong expression of CD19, we would like to see one or more of either CD10, CD22, or cytoplasmic CD79A. If the CD19 is a weak expression, then we need to see two or more antigen targets which are strongly expressed. However, I should point out, if T cell lineage is ever under consideration, you cannot use cytoplasmic CD79A as a potential target. So let's start off by looking at B-cell acute lymphoblastic leukemia. Now, morphologically, B-cell ALL can be quite challenging, with some as pictured here, and these images are from the American Society of Hematology ASH Image Bank. And you can see here, they do look completely different, so it can be quite diagnostically challenging using morphology alone. So this is a cartoon schematic of B-cell development and tolerance checkpoints, with the phenotype displayed below, from the pro-B cells within the bone marrow niche to the terminally differentiated plasma cell. And we know through multiple publications where antigens are gained and antigens are lost through B-cell development as it goes through central and peripheral tolerance checkpoints and become terminally differentiated B cells known as our plasma cells. Now, when we're looking at the subclassification of B-cell acute lymphoblastic leukemia, this is based on the EGL classification. We know that B-cell ALL should be expressing CD19, so there's our CD19 coming up positive across our subclassification of B-cell ALL. So pro-B-cell ALL, our very earliest form, lacks CD10, lacks cytoplasmic immunoglobulin heavy chain, the cytoplasmic M, and also surface immunoglobulin. Common B-cell ALL, we know that again expresses CD19. What we do see in common ALL is we get gain of CD10 or the CALR antigen, but again is negative for cytoplasmic mu and also surface immunoglobulin. Then slightly more differentiated precursor B-cell ALL, again has its CD19. It can still retain CD10, and we do see cases that retain CD10, or it again may be lost. But what we do see is a gain of the cytoplasmic IgM heavy chain, the cytoplasmic mu. So by having the cytoplasmic mu, that trumps if it has CD10, and these cases are pre-B-cell acute lymphoblastic leukemia. And there's again a reference to the publication should you wish to download it. This is a much more recent publication which has looked at the impact of immunophenotyping on the subclassification of B-ALL based on the EGL classification. For example, we know that those patients with a KMT2A, AFF1, the majority of patients will have a pro-B cell phenotype. However, it's very important that I say it's critical for the genetics to be carried out independently. Immunophenotyping can only support the genetic diagnosis. They go hand in hand together. So whilst we can see up here that KMT2A, AFF1, we can see that this is from a group in Tokyo. You can see here that the majority of these cells here have this pro-B cell phenotype, where fewer cases are the common CALR antigen expressed CD10 positive ALL, and slightly more cases, about 23% of cases, will have a pre-B cell phenotype. However, like I said, you can't use flow cytometry to predict the genetics, it can only support it. But it is quite useful and may be sort of advantageous and sort of helpful when we're looking, while we're waiting for the genetic studies to come through. And this is a table from that same publication which stratifies the phenotype against potential molecular subtypes. Now, the percentages of positive cases, which is more than 20% expression in the blasts of each antigen in each subtype of genetic abnormality of BCP ALL, is shown here schematically. Like B-cell acute lymphoblastic leukemia, T-cell ALL can also be quite challenging morphologically, as these cells here could easily do for, say, Burkitt or high-grade lymphoma. And I've seen cases of blastic plasmodendritic cell neoplasm look very much like T-cell ALL. So again, very challenging morphologically, which is why we need the additional investigations such as flow cytometry and genetics to actually define what these cells are. This is a little bit more classical, what we can see in T-cell acute lymphoblastic leukemia. So again, this is a cartoon schematic representation of T-cell development. So we know that those cases of pro-T-cell ALL may have CD34 on the surface. They will be expressing TDT. They'll have cytoplasmic CD3 expression. And we find that T-cell ALL has a very bright, strong overexpression of CD7. So if we have an abundance of these cells that are with a very overexpressed CD7 with cytoplasmic CD3, we know that these cells are most likely to be pro-T-cell acute lymphoblastic leukemia. As our T cells develop and they become early thymic, they can gain CD2. When we get to the sort of intermediate thymic stage, again, this is where we can see a gain of CD5, CD4 and or CD8. So for cortical T-cell ALL, we see an abundance of cells, again, that have that very overexpressed CD7. They retain their CD2, but then they gain CD1A. Then, of course, you have your late thymic T cells, where the CD1A is lost, but you can get a gain of CD3. And then we have our post-thymic CD4, CD8 single positive cells. So by knowing sort of T cell differentiation, and we know the process of pre-cortical to our late thymic and intermediate thymic patients, we can then define what T cell acute lymphoblastic leukemia stage they are. It should be also pointed out that T cell ALL can aberrantly express myeloid antigens. Quite commonly, we can see CD33, CD117, CD13 on our sort of ETP or early thymic progenitor T cell acute lymphoblastic leukemias. It's also important to point out that you can see cases of, say, pro T cell acute lymphoblastic leukemia that may gain a CD5, and that's what we call maturation asynchrony, where our leukemia has gained something that you might find would be expressed normally at the sort of intermediate or late thymic stages. So this is one of our older cases of TLL with a much more classical phenotype. So you can see here we've got CD45 versus side scatter in our first population. And these cells coloured in dark blue down here are our cells of interest. They're our CD45 negative or negative to dim population. And we've got our normal lymphocytes in bright green there. You can see when we plot forward scatter versus side scatter, they're all falling in that lymphoid region. Looking at the next plot along here, you can see that these cells are negative for surface CD3, so these are our normal T cells over here, and they're negative for CD56, these are our natural killer cells up here. So the cells that we're interested in are these blue ones down here. They're negative for CD4 and CD8, which is this plot here, so our single positive CD4s, CD8s, and it's negative for both. This next plot has got CD5 versus 23, and you can see that our normal lymphocytes have a stronger CD5. We can see that our population of our interest, our malignant population, has got a much weaker CD5 expression compared to the normal T cells. Again, they're lacking CD57 and CD8. They're lacking CD19 and 10. You've got a little bit of weak expression of CD34, but not much, and CD117 negative. Now, for T-cell acute lymphoblastic leukemia, a lot of the time we see that the cases are actually HLA-DR negative. Now, many people use HLA-DR as a potential immaturity marker in acute leukemias, but it's very important to remember that T-cell ALL, quite commonly we see HLA-DR is negative. And you can see here we've got CD13, CD33 and CD13, and our cells in blue are negative for both. Likewise, our normal T cells in green are also negative for both. So I think the key points here, we've got our CD, again, this is our extended phenotyping work. So you've got our gated population of blasts in blue, our normal lymphocytes in green. You can see here we've got cytoplasmic CD3 and surface CD3 on this axis. And you can see here you've got our normal population of T lymphocytes in green, which are co-expressing both surface and cytoplasmic CD3. However, our population of interest, our blast population, lacks surface CD3 but has expression of cytoplasmic CD3. I'm going to jump to this plot down the end here because this actually quite nicely shows our CD7 versus myeloperoxidase. You can see here we've got a very nice overexpression of CD7. Our CD7 on our blue population of blasts there is stronger than our normal T cells in green. So you can see here we've got cytoplasmic CD3 expression with an overexpression of CD7, and that's quite classic for our T-cell acute lymphoblastic leukemia. Very important to assess that this is not a mixed phenotype acute leukemia. So you can see that they're negative for cytoplasmic CD79A, negative for myeloperoxidase, and negative for 19. So I think the key points here are that the cells are CD45 dim. The blue ones are the ones of interest. We've got the normal T cells in green. We've got the abnormal T cells in blue, which are surface CD3 negative, but cytoplasmic CD3 positive, with an overexpression of CD7. We can also see that we've got TDT expression down here. And you can see, so we've got very nice TDT expression. And this plot here has got CD7 versus 99. And you can see that our malignant B cells in blue have a very overexpressed, strong expression of CD99. Again, quite characteristic of T-cell acute lymphoblastic leukemia. Most importantly, our antigens targeting the B cell lineage and the antigens targeting the myeloid lineage are all negative. So this was taken from Mike Borowitz's publication and is also referenced in the WHO, the World Health Organization guidance on hematological malignancies. And this basically, again, quite nicely characterizes our cells of whether they're pro-T cells, pre-T cells, cortical, mature, and then our ETP or early thymic progenitor, T cell progenitor, ALL cells there. So what we like to see is TDT positivity with, it can be quite variable expression of 1A, 2, 3, 4, 5, 8. We like to see cytoplasmic CD3 and CD7, an overexpression of CD7. And like I said, these can be subdivided into different stages by intrathymic differentiation. And it's a good read to have a good read of the Mike Borowitz paper that was published in 2008, and the reference is there for you to go away and read. Now, again, this is looking at this published study out of Tokyo that was published a couple of years ago, and this is a schematic schematic of the percentage of cases with corresponding antigen expression in T cell and T cell acute lymphoblastic leukemia, where the antigen expression is greater than 20%. So moving on to acute myeloid leukemia, and I'm going to just say here again that I'm going to go through the phenotyping of the subgroups in AML, and we're going to cover some of the morphology as well, where the morphology and the phenotype could show some correlation. However, it's very important that I say that the morphology and the phenotype should not be used as a substitute for genetic studies. There are only small parts. These are only small parts of the wider integrated diagnostics into acute myeloid leukemia. So where there may be some correlation, morphology and phenotype should not be used as a substitute for genetic studies. So let's look at acute myeloid leukemia. So this is AML with the RUNX1, RUNX1T1 fusion, the 821 fusion. Now, leukemic cells express a characteristic phenotype that includes a high-intensity expression of CD34, but we also see an aberrant expression of the lymphoid markers CD19 and cytoplasmic CD79A. You can see CD56 is positive in some cases, with expression on myeloblasts and maturing neutrophils. Neutrophils in these cases usually show abnormal maturation patterns as well, which is why it's very important to know the normal maturation patterns for your populations. Other markers commonly expressed include HLA-DR, CD13, and myeloperoxidase, while CD33 is usually weak or can even be negative. The blasts may show some maturation asynchrony, for example, co-expression of CD34 and CD15. PAX5 expression is usually absent. Now we know that those cases of the 821, the RUNX1 XT1 translocation fusion, we know that when we look at the morphology of the cells, the neutrophils can be some dysplastic, there can be some eosinophilic differentiation, but the blast cells usually have an indentation in the nucleus representing the Golgi zone, and there is often quite a long, thin sort of arrod, so sometimes there's a fusiform of an arrod, and you can see that quite nicely here, a very single, slim, slender arrod running through the cell. So within these plots on the right hand side, we've got the red neutrophils, the monocytes are in green, and this can be confirmed by expression of say CD14 and CD36, which isn't shown here, and you can otherwise be interpreted as degranulating neutrophils. In the cyan color, the blue color, we've got the blast cells, and in magenta we've got the lymphocytes. You can see that there are two distinct populations of monocytes and blast cells, although they almost sort of form a continuum from one running into the other. Now the blast cells are expressing CD34 and a weak CD33, as well as CD13 and CD117, but they're not expressing CD11b, which is what stains the monocytes and the neutrophils. So the blast cells and the monocytes and sort of part of the neutrophil population express CD56. And you can see there's also an aberrant expression of CD19 by the blast cells. And that can be done, like you can say, we just plot one antigen against side scatter, and you can pull out your populations of interest. So these are our blast cells down here with that aberrant CD19 expression on the surface. Something else which is a bit more of a clinical emergency is acute promyelocytic leukemia. Now, a distinction must be made between cases of acute promyelocytic leukemia with classical morphology and with variant forms. Classical APL has hypergranular promyelocytes, some of which may contain multiple rods, and also giant granules can also be present. The nucleus can be quite often bilobed, but because of the hypergranularity, this might be readily apparent morphologically. Then in the middle, you can see we've got the hypergranular variant and the microgranular variant, and then we've got the hyperbasophilic variant on the right-hand side. So immunophenotyping can be considered by some as unnecessary in acute promyelocytic leukemia. And I would somewhat agree. I think the most important diagnostic test in acute promyelocytic leukemia is obviously the PML-RARA. However, the phenotype is very characteristic, whilst sometimes slightly tricky. On the canonical sort of CD45 versus side scatter cartography, the first impression that may be the absence of progenitors or blast cells with large numbers of sort of slightly degranulating neutrophils, and that can be seen here. This might be easily confused with degranulating neutrophils. And in fact, this sort of flame-like image of cells with, and you can see here it's sort of like a flame from a candle. This flame-like cells with intermediate expression of CD45 reflects the granularity of these leukemic cells. So these cells will typically express the myeloid markers such as CD13. They usually have a nice bright homogeneous CD33 and are often CD117 positive. However, typically they lack CD34 expression. So testing for this sort of quadruplet of fundamental markers will thus strengthen the diagnosis of acute promyelocytic leukemia. We also like to see a very strong expression of myeloperoxidase as demonstrated by flow cytometry. So you can see here within our plots, we've got our normal T cells in the first plot. So this is our CD3 and cytoplasmic CD3. Our cells, that sort of flame cells that we can see here, have got a nice strong expression of myeloperoxidase. These cells are lacking CD19, they're lacking CD7, and they're lacking cytoplasmic CD3. So this is quite characteristic for acute promyelocytic leukemia. Now we know that cases of acute promyelocytic can have aberrant expressions of things such as CD2, CD64, and you can see here we've got a very nice plot of CD64 versus CD123, and you can see our blast cells up here are characteristically expressing both those two antigens. Now, the assessment of the cytological and immunophenotypic features enables this diagnosis to be made reliably and usually within sort of a few hours. And in the great majority of cases, this can be done before the confirmation of the translocations between chromosomes 15 and 17 by FISH or by conventional cytogenetic analysis. So the cytological features of AML with variant RARA translocations can be less distinctive. Cases associated with 1117, the ZBT-B16 RARA typically show a more sort of mature sort of maturation beyond the promyelocyte stage, with neutrophils being very dysplastic. However, ARODs can still be present. So this is a bone marrow film in AML with inversion 16, showing a myelomonocytic differentiation. There are two mature vacuolated eosinophils on the left-hand side in this picture here. One which has a non-lobed nucleus, and an eosinophil myelocyte has some granules with some basophilic staining, which is quite characteristic. And this is an MGG stain photographed under ×100. So in cases of AML with CEBPA MYH11 fusion, we quite commonly see two aberrant blast populations being identified. So you can see these multiple blast populations. One will be usually a CD45 dim immature blast population that expresses CD34 and myeloid markers such as CD13, CD117 and myeloperoxidase. Quite commonly you'll see a second, more mature monocytic, sort of more CD45 bright population that expresses monocytic antigens such as CD14, CD64 or maybe lysozyme, in addition to myeloid markers as well, but they will lack CD34 expression. You can sometimes find CD15 expression or CD65. Commonly they're CD11B positive, and they can have aberrant expression of CD2. So for our next case, we've got AML with KMT2A rearrangement. Now, patients presenting with this acute myeloid leukemia may have varying white blood cell counts. They can present with leukopenia, or they can present with overt leukocytosis, which may be up to 200 × 10 to the 9 per litre. In adults, the white cell count does not differ significantly from age-matched AML with normal cytogenetics, and no significant difference has been reported between patients with different KMT2A fusion partners. However, children often present with hyperleukocytosis and extramedullary involvement manifesting as maybe gingival hypertrophy or skin lesions is quite common. Now, KMT2A rearrangements are detected at all ages, with their frequency showing an inverse relationship with age. In children, KMT2A rearrangement is the most common genetic abnormality, constituting about 20% of AML cases. The frequency exceeds 50% in infants and about 10% in adolescents with AML. In adults, it's much less. The KMT2A rearrangements are found in about 2 to 3% of patients with AML. So the key points I think from this are obviously the peripheral blood features are that of acute monoblastic leukemia, sometimes acute monocytic or maybe myelomonocytic. If we look at the phenotype, this can vary based on the cell lineage. In children, we usually find a strong expression of CD33, 65, CD4, and HLA-DR, with a lower expression of CD34, 13, and 14. In adults, we see much more markers of monocytic differentiation with CD14, CD4, the integrin CD11b, CD11c, 64, 36, and 65. They usually lack CD19, and they can have varying expression of immaturity with CD34 and CD117 and CD56. However, you do quite commonly see a lower expression of CD13, and indeed CD7 has been reported in these cases. However, it's very important to say that there are no real strong morphological or immunophenotypic clues to the diagnosis of this AML. And these are just some of our scatter plots here to show of a recent case of KMT2A rearrangement, where we've got our blast populations falling here, population of cells that are CD117 positive, negative for 34, and then you can see you've got that separate population of 34117 progenitors. And indeed, these cells have a weaker expression of CD56, some weak CD4, a nice strong HLA-DR with a homogeneous 33, and a weak CD13, quite characteristic of AML with KMT2A rearrangement. This is just some extended phenotyping plots. You can see that these cells are CD38 positive, with a very nice strong expression of CD11c. Most importantly, they're negative for cytoplasmic CD3, negative for 19 and cytoplasmic CD79A. Nice strong expression of CD123, and you can see here again you've got that very nice strong expression when we plot CD64 against 123. These are our blast cells here with a very strong expression of CD64, and again this case you can see here if we plot CD7 against myeloperoxidase. These cases here we've got a very nice positive expression of CD7. Now this is a bone marrow smear from an AML with the NUP214 fusion, showing dysplasia of erythropoiesis, and also we've got dysgranular poiesis as well, again stained with a May-Grunwald-Giemsa stain. Now morphologically, the bone marrow is, if we look at the bone marrow, it's usually quite hypercellular. The bone marrow smears may show increased blasts with maturation or some myelomonocytic differentiation. In some patients, the morphological features are those of MDS with increased blasts, and multilineage dysplasia is quite common, including abnormal nuclear lobulation in the erythroblasts and myeloid cells, ring sideroblasts, and hypergranular myelopoiesis and micromegakaryocytes. In these cases of acute myeloid leukemia, the ALRODs are quite infrequent, and basophils may be increased in a minority of cases. So if we look at the immunophenotyping of these cells, they have a sort of a generic AML phenotype, and that means that they're usually positive for CD13, CD33, 34, 38, CD117, 123, HLA-DR, and myeloperoxidase. Now CD7, CD9, 15, 64, and TDT can be variably expressed, and CD34 may also be negative. So looking at acute myeloid leukemia with a MECOM arrangement. Now the peripheral blood smears often show dysplastic neutrophils with or without associated blasts. You can see red blood cell changes, which are, if they are, they're usually mild, without sort of teardrop cells. You can find giant and hypergranular platelets, as well as bare megakaryocyte nuclei being present. Within the bone marrow, the cellularity can be quite variable and can feature fibrosis. Now, the blasts in the bone marrow have quite variable morphology, often resembling those seen in AML without maturation, or acute myelomonocytic leukemia, or even acute megakaryblastic leukemia. Now, multi-lineage dysplasia is quite common and most pronounced in the megakaryocytes, which are often small and have non-lobated or bilobated nucleus. Now, if we look at the immunophenotyping of these cells are usually positive. These blasts are positive for CD34, CD33, CD13, CD117, and HLA-DR. Most cases are CD38 positive, and aberrant CD7 expression is quite frequently observed. Now, a subset of cases may also express the megakaryocytic markers such as CD41 and CD61. So looking at AML with the RBM15 MRT-FA fusion. Now, in these cases, blasts have quite a clear megakaryocytic differentiation. Now, the megakaryoblast that you see morphologically are usually sort of medium to large size with a round, irregular, or even a convoluted nucleus containing several nucleoli and quite fine reticular chromatin. The cytoplasm is often agranular, basophilic, and may also have that sort of blebby appearance. Micromegakaryocytes are quite common, but the dysplasia of the granulocytic and the erythroid lineages is usually absent. So any sort of potential dysplasia is predominantly within the megakaryoblast. Now, if we look at the immunophenotyping, the blasts of these cells and these megakaryocytes may express one or more of the platelet glycoproteins CD41, CD61, and or CD42b. Now, when you're staining for cells, you can surface stain for CD41, 61, 42b. However, it's very important to remember that cytoplasmic staining for CD41 and CD61 is much more specific and much more sensitive than staining for the surface targets. Now, CD36 is typically positive, but remember CD36 is not specific. Blasts can be positive for CD13 and CD33, whilst CD34, 45, HLA-DR myeloperoxidase, other things such as TDT and lymphoid markers are usually negative. This is another case of AML where CD7 has been also reported to be aberrantly expressed. So moving on to acute myeloid leukemia with NPM1 mutations. Now, most cases of NPM1 mutated AML are markedly hypercellular. If you do a bone marrow aspirate, it's absolutely full of cells. You see a myelomonocytic or a monocytic differentiation is quite common. However, the morphological features can vary quite wildly, with some blasts showing much less maturation than others. Multilineage dysplasia has been reported in cases of NPM1 mutation in about 20 to 25% of cases. Now, these cells are quite nice morphologically. These blasts have this sort of cup-like nuclear morphology, which are highly associated with the presence of NPM1 mutations. And you can see that quite nicely in some of these cells here. You can see they've got this very sort of cup-like nucleolus there. You could almost put a little handle on the side of that blast, and it could look like a coffee cup or a mug of tea. So, like I said, the key points are the hypercellular bone marrow with that myelomonocytic or monocytic sort of appearance to them. Of course, the patients are anemic, they are thrombocytopenic, and they commonly present with a high white count with this cup-like nuclear morphology in usually greater than 10% of the blasts. Immunophenotypically, they have a very strong overexpressed homogeneous CD33, and again these cells have a weak 13, CD117 and 123. If your laboratory tests for CD110, this is quite commonly expressed in cases of NPM1 mutated AML. Now these cases are typically associated with the lack of CD34 and HLA-DR independent of the cell maturation stage. A lot of cases where you find HLA-DR negativity, CD34 negativity, you might be thinking, well, that's quite characteristic of acute promyelocytic leukemia. It's very important again to look at the morphology, look at the cells. Do they have a higher side scatter that would be more in keeping with APL, or is it a high white count? Does it have that sort of cup-like appearance, myelomonocytic appearance, and does it lack HLA-DR and CD34 but have a weaker 13-117? That would fit more along the lines of an NPM1 mutated AML. Now, for AML with biallelic CEBPA mutations, there are no cytological clues to this diagnosis, although there have been some cases. I know Manelli et al. Reported a frequent association with erythroid dysplasia. However, the key points to CEBPA mutations are that they usually present with a higher hemoglobin level and a low platelet count and a low LDH. The smear preparations can show increased blasts with or without maturation, and dysplasia is quite common, especially dysgranulopoiesis and dysmegakaryopoiesis. Now, if we're looking at the immunophenotype of these patients, they again commonly will have CD13, CD33, 11B. They can express 15, 34, and HLA-DR. Now, aberrant expression of CD7 is found in between 50 and 75% of cases of AML with CEBPA mutations. Very importantly, these cases very rarely express CD56 or CD14. Now looking at acute myeloid leukemia with the BCR-ABL1 fusion, the morphological spectrum of AML with this is quite broad and can encompass AML with minimal differentiation, AML without maturation, or with monocytic differentiation. Most cases will express CD34 and HLA-DR, as well as our other myeloid antigens. However, aberrant expression of CD7, CD19, and CDT is not uncommon. But of course, it's impossible to diagnose these patients without our colleagues in the molecular services. And now looking at AML with MDS-related changes. Now, the diagnosis of AML with MDS-related changes requires that 3 of the following criteria are met: greater than 20% blasts in the blood or bone marrow, the presence of at least one of the following, which is a history of MDS or MPN, and 1 or more of the cytogenetic or molecular abnormalities that are listed. And you can see here I've got the WHO 2017 4th edition, the revision of it, and then the newly proposed WHO revised 5th edition that was published in Leukemia for AML with MDS-related changes. Most importantly, you need to have an absence of the history of exposure to cytotoxic therapy. You've got to have an absence of any other myeloproliferative neoplasm, and the criteria for myeloid neoplasms associated with germline predispositions must be excluded. Now, in cases of AML with MDS-related changes, blasts will often express CD34, 117DR, as well as our usual myeloid antigens such as myeloperoxidase and CD13. Now, occasionally in AML with MDS-related changes, you can find the expression of CD7 and CD56. Now, the expression of such antigens is usually quite aberrantly high or very, very low. The maturing monocyte, myeloid, and erythroid lineages can also show some immunophenotypic aberrations, which is why it's very important that you look at all your populations. You look at all your monocytes, your granulocytes, your erythroid lineages, to look at sort of abnormal maturation patterns. We know what normals are, so therefore it's important to look what may be abnormally expressed. Now, AML with mutated P53, like I said, there are two new classifications. We've got the ICC classification, in which AML with mutated P53 is recognized, but AML with mutated P53 is not recognized in the latest edition of the WHO, which is still in print within Leukemia, awaiting its publication in book form. It's important to note that AML with mutated P53, there is no specific immunophenotypic profile that would fit this. However, aberrant CD19, CD7, and TDT expression is very common. Like MDS, AML with a mutated TP53 is typically associated with sort of complex cytogenetic abnormalities and usually has a very poor outcome. Now, the WHO 5th edition that's been published in Leukemia refers to myeloid neoplasms post-cytotoxic therapy, and that includes myelodysplastic neoplasms. So if we look at AML, MDS, where there has been previous related therapy, this could be exposure to alkylating agents or ionizing radiation, those following exposure to topoisomerase II inhibitors. Now, the immunophenotype for these patients, the flow cytometry, is quite variable, and this really reflects the heterogeneity of these patients. However, you do usually see CD34, CD13, 33, and myeloperoxidase, although downregulation of any of these can be quite common. In addition, lymphoid antigen expression, including CD19, cytoplasmic CD79A, CD3, CD5, 7, and 25, can also be commonly found. So it's very important in cases of AML or MDS where they've had previous related therapy that we exclude mixed phenotype acute leukemia or MPAL. So that brings us quite nicely on to those sort of acute leukemias where you do have a mixed phenotype, or there is some ambiguity to the lineage of these patients. So acute leukemias of ambiguous lineage, very important to say they are rare. They're very rare. They're less than 4% of cases of all acute leukemia. What they do require is 20% of the abnormal progenitors that do not show differentiation along a single lineage. Now, this can include acute undifferentiated leukemia, where you lack a clear lineage commitment, but also includes something called mixed phenotype acute leukemia, or MPAL, where you see differentiation along more than one lineage. This may be T myeloid or B myeloid. Now, leukemias that can be assigned to another sort of well-defined entity should not be considered MPAL, despite meeting the lineage criteria below. So I think it's quite good here to put this slide up again that we showed earlier, which is how we can define cases of mixed phenotype acute leukemia. So we know that for the T lineage, expression of CD3 is the hallmark of the T lineage, either in the cytoplasm or less frequently on the cell surface. And a higher level expression is more strongly associated with T lineage, so an expression of exceeding greater than 50% that seen on normal mature T cells should be seen in a proportion of the leukemic cells. So if you have a case that has a nice strong expression of myeloperoxidase, which also has, say, a nice strong expression of cytoplasmic CD3, that could be considered a mixed phenotype acute leukemia of myeloid T. Likewise, if you have a nice strong expression of myeloperoxidase with a strong expression of CD19 with one other marker, say CD22, or more commonly cytoplasmic CD79A, this can be considered a myeloid B mixed phenotype acute leukemia. Now, when we're performing flow cytometry, it's very important that we use bright fluorochromes such as phycoerythrin, APC, or some of these more newer sort of brilliant or bright brilliant violet dyes that actually provide an increased dynamic range that aids in the determination of these cells. Now, when we look at the genetics, there's an increasing variety of molecular and genetic abnormalities that have been described in these cases of this ambiguous lineage, and often associated with particular lineages or immunophenotypes. However, immunophenotypic features in acute leukemia may change over time, or they can also change under the influence of therapy. In its most extreme form, this is seen with a single lineage acute leukemias such as ALL or AML, where you might even see an appear from a switch from one lineage to another. And this phenomenon has been particularly associated with such as the presence of KMT2A rearrangements, ZNF348 fusions, DUX4 rearrangements, and the PAX5. So if we take, this is just a table format showing these classifications where we can see these phenomenons of lineage switch, where at cases of KMT2A, we've seen MPALs with B myeloid, we've seen a lineage switch to AML. So this phenomenon suggests that these leukemias contain progenitors with lineage plasticity and may be biologically related to MPAL, but in fact these entities have been observed present de novo, such as MPAL at times. So maybe future further classifications may favor distinct entities defined by these underlying genetic abnormalities, with recognition for the variation that we see in the immunophenotype as a result of potentially second genetic events or the impact of prior therapy. So here we have some flow cytometry plots of one of our cases where we have found a mixed phenotype acute leukemia. And this is actually a very nice recent rare case that we have picked up through our service. Now you can see on the first plot of CD45 versus side scatter that we have our blast window falling down here in this green population here. And these cells are expressing CD34, so we're plotting here CD34 versus CD117, and you can see here we've got a nice expression of CD34. Now when we look actually on these CD34 positive cells, we can see that these cells are co-expressing weak expression of HLA-DR, a sort of weak to moderate expression of CD33, but are lacking CD13. If we then look at these plots down here, we've got cytoplasmic CD3 versus surface CD3, and we look at our blast cells that are expressing cytoplasmic CD3 but are lacking surface CD3. But more importantly, if we look at these cells in a little bit more detail, we can say that the expression of the cytoplasmic CD3 is at the same intensity as our normal residual T cells up here in blue, which are co-expressing both surface CD3 and cytoplasmic CD3. Now, if we look at these next two plots, we've got CD79A and CD19. You can see here that our blast population, our CD34 positive progenitors, have a weak expression of CD19 and a weak to moderate expression of cytoplasmic CD79A. But again, what is very important here is that the co-expression of these B cell antigens are significantly weaker than our more normal residual CD19 positive B cells shown here in blue. However, if we look at this last plot down here, we've got CD7 versus myeloperoxidase, and we can see here on this plot that the cells are co-expressing what's sort of like an overexpression of CD7. More importantly, not only are these cells, these cells, this mixed phenotype acute leukemia expressing CD7 or an overexpression of CD7, we can also see here that they're co-expressing myeloperoxidase. Not only are they expressing myeloperoxidase, we've also got this very nice overexpression of CD99. So this is a very interesting case, like I said, that has there's no clear one lineage associated with it. So what we have here is because of the no clear lineage identity, we've got a mixed phenotype acute leukemia that has CD34, 33, lacking 13, with a cytoplasmic CD3, myeloperoxidase, an overexpression of CD7. So this is what we would call a MPAL mixed phenotype acute leukemia of T myeloid, But we must also remember that these cells have also got some aberrant B-cell antigen expression, where we're seeing weak CD19 and a weak or weak to moderate expression of cytoplasmic CD79A. Now, this was actually a case who was an MPAL T myeloid at diagnosis with very weak expression of CD79A. They were then post-transplant MRD negative. And their relapse phenotype is this mixed phenotype acute leukemia, or MPAL T myeloid, which has now gained sort of like a weak to moderate expression of cytoplasmic CD79A, and has also gained weak expression of CD19, which wasn't there before. So I thought I'd finish up with just a case study to just show how everything integrates together, how you need not just flow cytometry, not just morphology, not just immunohistochemistry, not just genetics, to come up with an integrated diagnosis, an integrated approach to patient diagnosis of acute leukemias. So this case history is a 36-year-old male who was rushed to our emergency department having felt unwell. With the following hematological parameters, with a hemoglobin of 100, white cell count of 81, and a low platelet count of 31. They did a bone marrow aspirate, which was inadequate but particulate due to the poor quality of the smear spread. So the cellularity was quite hemodilute. The megakaryopoiesis was decreased, but with normal morphology, and erythropoiesis was reduced. Granular poiesis was markedly increased, and the blasts were increased, and different when a manual diff was performed, came out at 88%. Some of the cells seen had cytoplasmic inclusions suggestive of hemophagocytosis, and you can see the differential there of the bone marrow. 200 cells were counted with 88% blasts, with monocytoid cells seen. So this is the morphology of some of those cells. You can see here we've got typical blast cells there. Some of them looking at the morphology, might give away what the underlying disease is, but you're certainly looking at an acute leukemia. Here's some more of these cells. Again, some of them quite similar, some quite homogeneous in nature. So of course the cells are sent off for flow cytometry, and we've looked at the consensus result of the cells here. So the positive antigens are CD4, 11B, 11C, CD13, 14, 33, 36, 38, 45, 64, and DR. So immediately we can say that these cells have got some sort of monocytic differentiation with CD4, our integrin CD11B, 11C, and our myeloid antigen CD13, CD33, and also CD64 and 14, again pointing towards that monocytic differentiation. Most importantly, the negative antigens for CD2, 3, 5, and 7, and 10. So obviously looking at some of our T cells there, CD19, 20, the negative for CD34, so our stem cell factor, our immaturity marker, negative for 56. These cells are also negative for CD117 and also negative for myeloperoxidase. So if we look at our lineage associated antigens, we know that myeloperoxidase is negative, which is what we like to see positive in acute myeloid leukemia. CD19 is negative, and CD7 is also negative. So no evidence of T cell or B cell, but we've got certainly a monocytic differentiation when we look at the positive antigens. So our colleagues in the genetics lab have looked at, done some FISH analysis. They've looked at 100 interphase nuclei, which showed no evidence of MECOM or the KMT2 rearrangement. However, the molecular genetics did find a 4 base pair insertion of NPM1 mutated AML, pardon me, with also a FLT3 ITD with a high allelic ratio. Now, our colleagues in the histology lab had a single core of bone marrow to fine measuring 12 millimeters, showing an increased cellularity, about 90% cellularity, so quite hypercellular, with occasional and disturbed erythropoiesis. Granular poiesis was increased greatly without maturation, with this monocytoid appearance. Thrombopoiesis was not identified on H&E, and reticulin stain was normal. So when we're defining cases, we say, what's the lineage of the cell? Is it lymphoid? Is it T cell? Is it B cell? Is it myeloid? And the lineage is certainly myeloid, and it's going down the monocytic sort of lineage. Is it mature, immature? Well, we know that they're lacking CD45, although they don't have the immaturity markers of CD34, CD117. We know that these cases of NPM1 mutated AML quite commonly lack CD34 and CD117. So the full diagnosis by the WHO would be an acute myeloid leukemia with NPM1 mutation. So that just shows the integrated approach that we need to have for these acute leukemias, in which flow cytometry just plays a very, very small part. So let's cover some of the limitations of flow cytometry. Sample quality and preparation. The analysis will only ever be as good as the preparation. We know that submalignancies, so not just in acute leukemias, but in all hematological malignancies, if you've got a very poor pull of the sample, or it's quite a fibrotic sample, or if you've got a case where the sample is quite clotted, it may not give you an accurate representation of what is going on. Flow cytometry has to have quite experienced operators who actually have to understand the principles of not only operating the system, but actually having a good understanding of the biology of the disease. You need to have good quality control, good quality maintenance. And then there's this argument of whether you should be using control samples to set your gates, or do you use internal biological negative controls? It must always be used and combined with morphology, because the use of single cells in a suspension, like I said, does lead to loss of tissue structure. And it can be quite costly. These monoclonal antibodies that we buy from commercial companies can be quite expensive. And you can end up producing a number of panels to diagnose these patients. And like I said, standardization, whilst we really want some standardization for flow cytometry, and really the Euroflow group has been leading on this, laboratories are still choosing to do their own panels and sort of picking the best bits of best practice guidance. However, what are the pros of flow cytometry? Well, it can be quite quick, and it's often used as sort of an instant screening process. When we talk about instant screening, we can tell whether it's B cell, T cell, myeloid, whether it's a mature disease or an immature disease. And then that can direct the subsequent testing of our colleagues in the genetics labs. It truly is multi-parameter. We can combine lots of colors, lots of antibodies with fluorochromes, so that we can actually really identify what these cells are. This allows us to have increasing sensitivity and specificity, which is really good for when we're looking for a measurable residual disease, or MRD. It's also very good where limited sample is available and can be used with a variety of sample types, provided those cells are in a suspension. So really, to summarize, flow cytometry does play a really important part in acute leukemia diagnostics, but it is alongside traditional investigations and more specialized investigations such as cytogenetics and molecular testing. Like every other method of diagnostic pathology, flow cytometry does have its strengths and its weaknesses. However, the speed of flow cytometry will help guide the diagnostic process. You have to be very careful not to overinterpret flow reports and flow plots. And it must always be reported in an integrated manner with morphology of the sample that you're performing flow cytometry on. Now, obviously, we are seeing a lot more development in the hardware and the technology, which has really sort of helped improve the diagnostic technique. And integrated diagnostic services have been developed in many centres in many countries, which is essential in providing the right diagnosis to the right patient to ensuring they're getting the right therapy. Now, this was taken off of Twitter. This is just a diagnostic algorithm, which you can pause on this slide, which is quite nice, which basically looks at some of the newer classifications, the 5th edition of the WHO and the ICC classifications, looking at AML classification according to genetic analysis. So finally, I'd just like to say thank you very much for listening to this presentation. And I hope this has been beneficial for you and has sort of given you an insight to how flow cytometry is an important sort of small but important part to acute leukemia diagnostics. Thank you very much.